Welcome to Valley Fever Voices podcast. I have Dr. Galgiani here, and Dr. Galgiani received his MD from Northwestern Medical School and completed a fellowship in infectious diseases at Stanford. In 1978, he joined the faculty of the University of Arizona, where he has remained ever since, currently as professor of medicine. Dr. Galgiani has focused his career on the special problem of coccidioidomycosis, also known as valley fever. His work has included studies of the impact of valley fever on the general population and on special groups such as organ transplant recipients and patients with AIDS, and is the lead author on the Infectious Diseases Society of America's practice guidelines for coccidioidomycosis. In 1996, Dr. Galgiani founded the Valley Fever Center for Excellence. In collaboration with Banner Health, Dr. Galgiani developed and is now implementing a training program for primary care clinicians for prompt recognition and optimal management of patients with new valley fever infections. The center's research has recently developed a vaccine candidate to prevent valley fever in dogs and potentially in humans. Thank you for joining me, Dr. Galgiani. This is going to be a wonderful talk.
Well, thank you very much for inviting me, Pamela. So I did couldn't resist putting together a little presentation, but this should be
Fabulous.
informal, and
Excellent.
and I'll set that up in just a moment. But feel free to butt in as much as you want because it's much more fun that way.
Yeah, I
And
have lots of questions, and I can actually pronounce coccidioidomycosis, which is a bonus.
you can actually say coccidioidomycosis. In fact, our student who is now staff at the Valley Fever Center, Zoe, she created a T-shirt that we gave to new members of the center, which says, say this word 12 times fast, coccidioidomycosis, 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 so forth. It's a great T-shirt.
It's wonderful. I know it's the craziest word in the world, but hey, we've got it.
Well, it has a derivation. Somebody, the original patient with this infection was in Argentina. Original
Uh-huh.
one discovered, at least recognized medically. That was in 1893, and it was thought to be a protozoan infection.
Hmm.
But the histopathology, the tissue reaction was sort of granulomatous. So they called it a coccidioidal granuloma because it looked like coccidia.
Uh,
And and then a few
it
years
gets
later,
confused
after it
with.
was just, pardon me?
It's confused with that when you
Yep.
talk about it.
Yep. And then a few years later, it was discovered in the United States by some people at what was going to be Stanford in San Francisco, and then one of the pathologists discovered it really was a fungal infection and not a protozoan infection. So they changed the name to coccidioido or coccidia-like mycosis. So that's where it came from.
Makes sense. Yeah. Yeah. I always wondered why it was so close to coccidia because that gets confused in veterinary medicine a lot. People think you're talking about an intestinal parasite. And I'm like, no, it's a systemic parasite.
So let's see. Can you see my screen now?
Yes. Oh, there we go.
Great. So I'm going to just go ahead and talk a little bit about for some of your viewers, your podcast listeners are not going to know as much as you do about valley fever. So I thought I'd give an orientation to what this really is.
That would be
So
fabulous.
yeah, we've already agreed it's a fungal infection and it's caused by the genus Coccidioides. And it used to be always one species, Coccidioides immitis. But it's been discovered that genetically there are two clades or two branches, and one's now still called Coccidioides immitis, and the other is now called Coccidioides piscidacea. They didn't really discover a new species; they just took the disease and split it into caused by two different clades, and call them two different species. And what's really interesting about that is that imidis is found in California. It's actually found up in southeastern Washington a little bit, and but it's pretty much limited to those areas along the West Coast. Coccidioides piscidacea it comes in everywhere else, including Arizona, and the winds blow back and forth between California and Arizona, and yet this geographic separation of these two different genetic differences stays pretty stable, which really speaks to the idea that probably there's a lot more to the ecology of the infection rather than wind blowing around.
Yep.
Yep. So it turns out that mice are very susceptible, at least some mice are very susceptible. You can be lethal with a single spore in mice. And we think that probably most infections in other mammals, including humans and dogs, is caused by single spore exposure. You can have high inoculum, but that's in special situations. Like there are great examples in archaeology digs, for example.
Mm-hmm.
And the CDC now estimates there's a much higher rate of infections per year than is reported. There's about 20,000 or 22,000 infections per year that are reported to the CDC, but the number is probably much higher than that goes unreported for a variety of reasons. And weather is probably a very important factor for the rates of infection, and there's a pretty elegant climate model suggesting that over the rest of the century, that the endemic region for valley fever may reach all the way up to the Canadian border.
Holy cow.
Yeah, so we'll see if that pans out, but that's based on temperature and rainfall predictions for the next 80 years.
So
So it turns
those
out
different species are related to the geographical area?
that's right, and it's very stable. So in fact, if you get into it, the genetics of the species that live in the Phoenix area are demonstrably different than most of the isolates that come from even Tucson, which is only 130 miles
Wow.
away.
Wow. Lots of stuff
But,
we need to know about.
right. So the spectrum of disease goes from a whole lot of patients who get infected but never know it, and they just don't get sick, or at least sick enough that they need medical care. But then about a third of patients who do seek medical attention have typically symptoms of pneumonia and a variety of other things that last for weeks to months. Now, they get better on their own. So it's a self-limited infection. We know that because there wasn't any treatments for valley fever till the 1960s. So there's a lot of natural history data. But this is anything but trivial. The disease in humans lasts, the ones that seek medical attention last a lot longer than people like. So it's not 2 or 3 days, but as I say, many weeks or even many months. And then a small percentage of patients go on to have it spread from the lungs to other parts of the body through the bloodstream to, say, the brain or bones or skin. And those are not self-limited. Those are very serious kinds of infections.
Mhm.
And they get to be good friends. Those patients get to know their doctors quite well because they often need years of treatment. And patients with meningitis, actually, it's recommended that they never stop treatment. It's lifelong treatment from that point on.
Serious stuff.
And yeah, and the meningitis specifically is uniformly fatal if it's untreated. So it can go from causing no disease whatsoever to fatal outcomes.
But not
And
everyone gets to that point. Only a small percentage of people get to the disseminated point.
that's right. Fortunately, that's a small percentage, but if the numbers of infections is pretty large, even that small percentage gets to be real numbers.
Mm.
And it's very concentrated. I'll show you some maps in just a moment, but it's not spread out uniformly across the United States or the Western Hemisphere, but is concentrated in certain areas. And economic impact that's been done for California and Arizona combined suggests, and this was several years ago, something like $1.5 billion overall economic impact. So and that's concentrated in certain areas and certain groups of elderly people, people of color, pregnancy, those that have profound immunosuppression are in particular risk for this. And if you run the numbers, the in those areas where it's endemic, the impact on the individual person, per capita impact, is very similar to that for polio, chickenpox, or measles before they had their own vaccines.
Wow.
Yeah. So I mentioned that many patients get over this without any treatment. And it turns out that those patients then never get it again. If they inhale another spore, we don't know about it, they never get sick from it. And that implies the idea that maybe we could do the same thing without having to wait for an infection, but to get protection through a vaccine strategy. And I'll get back to that towards the end of this discussion.
Excellent.
So this is a picture of where I live. That's the front range of the Catalina Mountains behind Tucson. And in the lower left-hand corner, can you see my pointer, by the way?
Yes, we can see it.
Great. Yeah, this is a scanning EM of the Arthrochonidia. That's the spore that if you looked at that on a petri dish, it would look like bread mold. It's a
Wow.
mycelial elements, but each of these little white things here, this is one single spore. Turns out the mycelia, which grows by elongation, as it dries out, every other cell becomes autolyzed and very fragile, but the ones in between stay viable, and those get into the wind. They're about 3 to 5 microns in diameter, and they go on to get in the air very easily. That's about the same size as a mycobacterium that causes TB. So they get it. They're a very good delivery device to get into the lungs. And if it does get into the lungs, whoops, you get a pneumonia. This is a X-ray. This, of course, is the heart. That's normal.
Hmm.
But this little thing over here is a pneumonia. And this particular one, there's no label on it, but that one's caused by coccidioides. And then if you looked at that under a microscope, you'd see these very large structures. I mentioned the Arthrochonidia is 3 to 5 microns in size. This is 70 microns in diameter. Each of these little spots here is a single white cell. So these are very large. And inside is maybe 100 or so progeny that are called endospores. And when these spherules, which is what the big, large spherical structure is called, once they grow and mature after 3 or 4 days, they rupture and spill out 100 times as many live progeny as starting with that first one. So the incubation time for humans is 3 to 5 um, weeks, or 7 to 3, 1 to 3. It can be as early as 1 week, I guess, and 1 to 3 weeks is really the incubation time. And so that you can see after several of these multiplications, you could easily have a good inflammatory response to it. So here we have a bar graph just showing you annual reported numbers of cocci patients. And yellow is for Arizona, and red is for California. And these, between the two states, is probably over 95% of all reported infections in the United States. And in general, Arizona has been about two-thirds of that compared to California. And here is a picture of my state, Arizona, showing you the intensity of where the infections occur. And you can see, if you know the geography and the roadmap of Arizona, there's an interstate that goes between Tucson and Phoenix. And that's pretty much the Valley Fever corridor because two-thirds of all U.S. infections occur up and down that stretch of about 150 miles.
Wow.
Pretty
California's
intense.
catching up though.
Yeah. So if it were to become like that across the rest of the United States, it would no longer be a small orphan disease. And people, it turns out, you may know, they travel in cars and planes and things.
Mm.
And this is a very pretty map that Caitlin Benedict published a few years ago. And it shows you where these brown spots are where valley fever was diagnosed out of the endemic region, and then histories of where they had been recently. And you can see a lot of people had visited Bakersfield, Central Valley of California, where valley fever gets its name, and also the area around Phoenix and Tucson in Arizona. And over here, you can see this is the map. I noted this is a newly discovered area around southeastern Washington that has the fungus in the soil there. That's been recovered there. And then there are other little pockets here in northeastern Utah. There's a national monument that's recovered valley fever as well. So it really is this area, but parts of it. And I've been talking pretty much about humans because that's my training as an internist in medicine. But there is a problem with pets as well. And Lisa Schubitz, who's shown here, a veterinarian at the Valley Fever Center, has demonstrated that the dog rate of infection is 6 to 10 times as high as it is with humans. When these dogs get valley fever, 25% of them have the bad form of the disease, whereas it's much less common in humans. And in Arizona alone, she estimates that maybe 30,000 dogs per year get diagnosed with valley fever. And for her and for the veterinarians treating um, dogs with valley fever, the standard of care is to treat all dogs. And she and Chris Berkowitz and Dr. Sykes and as I say, Lisa Schubitz herself have just submitted for publication an estimate based on skin tests, I'm sorry, on cocci diagnoses across 6 different states, and come up with an estimate in 2024 that the cost to pet owners is 80 to $100 million per year.
That's
So it's not,
insane.
yeah,
Yeah.
and it's a real impact on pet owners. We're talking about vaccines, and if there were a vaccine, and we have an idea that there may be for valley fever, that would be a huge step forward for this disease.
Yep. And pet owners really don't have insurance for the most part for the dogs. Like with people, they have medical insurance, and that's a problem for our dogs.
That's absolutely right.
It's coming out of pocket.
In their publication, they discuss, discretionary income, and if you have a dog who gets sick with valley fever, that one dog, and this is only they only did the modeling for the simple, uncomplicated forms of valley fever for dogs, and it doesn't take some really very expensive management of the most seriously ill dogs, and just for that alone, it would be a big chunk of disposable income that would just be swept away for that one
Yep.
little problem.
Yeah, it could be tens of thousands of dollars for dogs, like if they get it in the pericardium around the heart and have to have surgery and all that. It's tens of thousands of dollars for surgery.
That's right.
Yeah,
And as you well know, not everybody can afford that, in
right.
which case choices have to be made, and dogs are often given up for adoption just for that, or have to be put to sleep.
Yeah, and that's where dog insurance or pet insurance comes in handy for those cases. I have a few of my clients that have had pet insurance and were able to afford that kind of thing. So fortunately, they're out there.
Do you have to get authorization for all of that stuff for pet insurance like you
No,
do for human users?
yeah, no, we don't do pre-authorization. They
Well, that's
they
good.
just submit the receipts and hope for the best.
Well, that almost makes me think I should have gone into veterinary medicine.
Yeah, well, it has to come out of pocket first, and then they get reimbursed in a lot of
So,
the.
yeah. So I mentioned that this whole idea of a vaccine a couple of times now, and it turns out that there's a valley fever vaccine that was discovered here at the University of Arizona several years ago, and it's really a wonderful One Health story. It turns out it starts with a gene in the fungal genome of a fungus called Cochleobolus, a fungus I only know because of this story. The gene's name is CPS1, and the fungus Cochleobolus is a pathogen, but it attacks corn, not animals. And research into Cochleobolus, where they did what's called targeted mutagenesis, where they randomly inject a piece of DNA into the fungal genome and then look to see what the effect of that is on the mutant. And if they find interesting effects, they go back because they know the sequence they stuck in the genome. They could find out which gene that was disrupted was responsible for that change in the clinical appearance of the fungus. And they found one that seemed to reduce the ability of cochleobolus to attack corn. So Mark Orbach, who's a good friend now of mine, a fungal geneticist, who had originally worked in phytopathogen work in plant and fungi that attack plants, and has now gotten a lot more interested in coccidioides. He found the same gene, or at least a similar gene to CPS1 that was in cochleobolus in the genome of the fungus coccidioides.
Wow.
And knowing that, he then decided, what's going to happen if I take that gene out of the coccidioides genome? So he did that. So he actually developed a procedure to do the genetic manipulation to do that. And so he basically snipped out that gene, which gives it the name delta, or subtracted CPS1, or delta CPS1 is the name of this mutant. And what he found was that that mutant actually was unable to cause disease in mice. And by unable to cause disease, I've mentioned 1 to 3 or 4 spores is enough to kill a mouse, or at least the mice we use in our lab. 25 million spores caused no disease in those mice, which was pretty amazing. And then mice that have been genetically engineered to have no immune system, they're called SCID mice, or severe combined immunodeficiency mice. They didn't get sick if they were infected with this mutant either.
Hmm.
And there are many other studies along the line of that with other mutants of mice, and also in pigs and in dogs, and now also in rabbits. And there's no suggestion that this mutant is able to cause disease after infection. And it seems to be, we call it first generation incompetent. That means if you were to introduce the fungus into the animal, it's alive, but it doesn't go through that full cycle going to a mature spherule. It falls apart before that happens and doesn't propagate. So I'm a physician, and that is important to me, that it looks extremely safe. And I think that's a very important factor. And it does, however, need live spores. And we've shown in various ways that if you inhibit or kill the fungus, it no longer confers protection. It seems like it needs to grow a little bit, and maybe in that way express or show an antigen to the host immune system after the infection to trigger the immune response to produce the protection. And as I mentioned, it's very safe. And I'm not going to show you all the data, but there's enormous number of studies done in mice and and safety also in other species. And the efficacy in the mouse model shows that it's better than any past vaccine that we've ever had come along. And there's been interest in a vaccine really since the 1950s. I've been involved, actually, there was a clinical trial in humans in the 1980s, which was a very effective study, and demonstrated that that vaccine should go no further.
Oh,
So
wow.
it was very convincing that that vaccine had no future. But there's just an example of what we've been able to do in the past. But this vaccine looks much more protective, and it was made in the mutant was made in the Coccidioides piscidacea. But if you challenge mice that are vaccinated with the imidis strain species of Coccidioides, you get cross protection. So it would work either way.
Would this be a modified live vaccine then?
That's right.
Okay.
It would be like a measles vaccine
Yeah.
in that
Okay.
sense. That is also a live vaccine. Possibly, and this would have to be proven, but possibly because this is incompetent in propagating. So the measles vaccine and most attenuated strains are recommended not to be used in people that are immunosuppressed.
Mhm.
In fact, that's part of the problem with not having a herd immunity in a population, because there are some people that just can't get the measles vaccine, right?
Mhm.
And if you have measles in your community, those people that can't get vaccinated are at major risk of that infection. But with this vaccine, since it doesn't seem to be able, even in immunosuppressed animals, to be able to uh, cause disease, It's like it turns out a new vaccine against smallpox was made that's called first generation incompetent. And its labeling doesn't forbid it to be used in people that are immunosuppressed. It's that safe. And it's very possible we would find out that this vaccine could be just as safe.
Nice.
So it is live, but I think because because it is live, the best vaccines in the world have always been live vaccines. And measles, for example, that vaccine, after you get 2 doses, you don't have to get boosters. It's lifelong protection, right? And so it's very possible, since valley fever wild type infection gives you protection for the rest of your life, that this vaccine, also being a live vaccine, might do the same thing. We don't know that yet, but it's a potential benefit of this strategy. And then it also, as I said, protects against immitis, so it's cross-protective. So what do we do next? Well, it turns out that the NIH had a special grant proposal, or request for proposals, to develop vaccines for small markets, and they required it to be submitted by somebody that had a commercial partner for that submission. So we partnered with a company called Anavive Life Sciences, which is a veterinary company, and we proposed, this is now several years ago, to make a vaccine to prevent valley fever in dogs. And the company thought this was a very interesting idea because they could then make a vaccine to be a veterinary vaccine. The NIH is really not focused on veterinary vaccines. It's much more interested in human health. And the argument we made to the NIH was, well, if we showed it protected dogs, that would be proof of concept that we should keep going to humans. And so that argument sold, and they actually funded that grant. And we, working with Anavive as our partner, then proceeded to demonstrate that this vaccine was protective in dogs. And I'm showing you just one data slide. This is a study of our vaccine. And over on the left-hand side here was the vaccine doses, 10,000 spores given both prime and boost. That's what PEB stands for. 50,000, 100,000, or 100,000 only as a single dose, as a prime only. Those sound like large numbers, but 10,000 spores is less than 1 microgram in weight. So it's a really small amount of stuff. And then after vaccination, a month later, animals were infected with a large number of spores by a respiratory route. And on the left-hand side shows you a composite disease score. And you can see those animals that didn't get vaccinated at all had a fairly high disease score. And this is composite of symptoms and a variety of other chemistry labs and other things. And then the prime and boost, these three here are essentially the same. And look how different this disease score is, how much lower it is. The 100,000 as a single dose is sort of in between. And it suggests that probably 2 doses is more effective in making a protective vaccine than one in this study. But all of them showed protection against the challenge in this model. So we were very excited about this data. And so where are we now?
Even the low dose had protection.
Yeah, and this dose of 10,000 spores actually is the same optimal dose in mice.
Yeah.
So even though dogs are bigger than mice, the same dose was just as effective as is in a mouse. So it's not unlikely that even in humans, we might not have much higher dose than that. That needs to be determined. But maybe the small dose is enough to do it.
That'd be nice.
Yeah, and the idea of lower is better because the local reactivity depends on dose. So how sore an arm you might get. And in fact, the local effects were relatively modest. So here we are. As I mentioned, this grant was awarded to the University of Arizona with our partner, Anavive, and it is in the process of taking these preliminary data and has done extensive work since then to create a proposal to the USDA for an application to make this vaccine a product to be used by veterinarians. And we're hopeful that it might be available as early as the end of this year.
Wow.
Things sometimes don't go as fast as you like, it turns out. But that's our hope, and maybe next year. But in the near future, we may have a vaccine available for dog owners, which is pretty exciting. And
That's
then, go
fabulous.
ahead. Yeah, that's really
It's been in the
exciting.
works for decades though, hasn't it?
The idea of doing this has been
Yeah.
around, yes, for over 60 or 70 years. Getting it done is another thing. And then last year, Anavive was awarded a contract from NIH that could be as much as $34 million dollars to take this vaccine further from where it is now and move it eventually under this contract into a first human trial. So it's now not just an idea, but there's some reality to the idea of taking this not only into a veterinary approval, but also into humans.
That's
So
exciting.
so our tagline is let's protect both ends of the leash. And that's kind of this One Health kind of idea. And the idea that making a vaccine for a dog use and then to humans, there's actually lots of examples of that. Uh, and it's been quite a ride for me. I've learned a lot about how you prepare vaccines for veterinary purposes as well as about this vaccine in general. So
That,
that's the story. I thought I'd pitch to you and let you people say and see
yeah.
if your audience liked it.
Wow. Yeah, that's exciting. So the vaccine would be for patients, dogs first, that do not have the disease. So
Great.
do not have symptoms, do not have the disease. It's a preventative vaccine. It's not something we're going to use for treatment.
Right. In fact, we have some data. We didn't ask that specific question, but we wondered if if it would be more it would cause any side effects in infected animals. If you vaccinated infected animals, would they get anything that uninfected animals would? And the answer is no. At least thus far, it doesn't seem any more dangerous to vaccinate some animal who'd already been infected. And so, they also seem to get no benefit either. So I think we don't have much. In fact, we think they're vaccinated by the infection itself. So and unfortunately, they have some consequences of that, but they probably have developed some immunity.
And so dogs in endemic regions would be vaccinated. Is there any other um, group of dogs that you would think would need a vaccine? Or are we just dealing with endemic states right now?
Well, as I say, not only do people travel, but sometimes they take their pets with them.
Hmm.
And kennel shows and
Oh, yeah.
trials and things that, like in the wintertime, to go to sunny places like Arizona. There might be a big interest in protecting show dogs from getting infected if they happen to go to a show that was held in Phoenix.
Wow. Um, so that would be for traveling dogs. So we would recommend the vaccine for dogs that are going to travel to states that have valley fever. Wow. That's
Yeah,
huge.
yeah, and I think there's lots of reason to think this will work. The data looks very solid. And as you know, there would be a interim period after preliminary approval to monitor injection sites for the next 1 or 2 or 3,000 dogs that get infected or get vaccinated to make sure it's well tolerated. We
Hmm.
expect it will be, but that hasn't been uh, demonstrated in that many of dogs yet. But other than that, I think there's no reason to not think it won't be available. And I'm very excited about the idea it would also be useful for humans.
That's great. I think a lot of people would be interested in preventing it. The people that know about it now and are traveling to the West are really nervous.
Right.
Yeah.
So
So
another thing I might just sort of point out, since I did um, find I was the founder of the Valley Fever Center almost 30 years ago. Um, this vaccine um, didn't happen on its own. It was Mark Orbach, who was the co-discoverer with Lisa Shubitz. He was interested in how the fungus causes disease. He had a basic question about pathogenicity. And that's why he took that gene out of that Delta CPS1 mutant was created. He wasn't exactly looking for a vaccine. He was looking for fundamental biology. But out of that search fell out this vaccine. And it it's hard to imagine that gene being identified. It was rationally discovered because Mark was looking at what causes disease.
Hmm.
And so it's a rational, it was not like by accident he found that gene. But once he found that gene, the consequence of that was that he found a vaccine. And and that happened because we have a Valley Fever Center for Excellence. And that center pretty much, we get grants, which is very valuable. But underneath that, the floor of the center is primarily by people who want things done for Valley fever. And
No.
what was preliminary data that we generated was not from grants, it was from donations from pet owners
Oh,
primarily.
wow.
And and I'd like to think that people who want to see something done about valley fever for humans also would want to support the center. So I just
That would be great. So I'll put the link for the Valley Fever Center for Excellence in the show notes below
right.
this video so that people can check out the work that you're doing with vaccines and maybe become a sponsor or a promoter.
Right. The pretty exciting thing, and you have to be careful because this disease is very addictive. There's many things we don't know about it because it's so sort of regional. There's been much less work. It's not an overpopulated area of research. It's not like like cancer or heart disease or and the result, you can ask a question, you suddenly find out that's a really interesting question. I wonder what the answer is in many different directions. And the implication of that is, if we knew more, there are so many things we could do about this disease, managing this disease so much better, not only a vaccine, but better treatments, better diagnostics, better understanding of who gets bad disease and who doesn't, and why they get bad disease, and how we could change it for them not getting bad disease. And all of that is within the purview of our center. So we'd love anyone who wants to see this as something they want to be a part of. We'd love to have them join
Yep.
us.
Yep. There's a lot of research that needs to be done for sure.
Yep.
Yeah, that's great. Are you seeing any side effects or adverse reactions with the vaccine besides just pain at the injection site?
No, we haven't seen any. It's looked quite safe. And part of the reason that would make sense is that infections with the vaccine, wild type infections, don't have much autoimmunity or things other than the real things that the infection causes. We know a lot about what happens with wild type infections, and 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 I think this is a live vaccine is likely not to cause any more of side effects than a wild type, um, and and it won't cause disease.
And does it protect 100% or at least close to that of dogs that have gotten the vaccine? Or do we have some slip by and get the infection anyway?
It's not 100%, and the question is whether or not all the dogs got a take. And we don't quite know the answer to that yet. It's not, we don't have enough data. It's
Mm.
very effective. Most mice are protected, and we've done that experiment a lot of times. And so and we actually now have some, this is now in press in a veterinary journal. We now have tools to study dog blood to identify whether or not they've been infected in the past, and also we can demonstrate whether or not they've been vaccinated. So I think we
Oh,
now have a way to find out if you vaccinated a dog and wait a month and did a blood test, we could probably say this dog has got an immune response to the vaccine, and then we would we could find out if that dog then would never get infected through studies. And my guess is probably those dogs will be protected. We might find that some dogs don't get a take, and that's a separate issue, but we'd be able to determine that.
Wow. So can you just distinguish between vaccine protection and previous exposure protection when you're looking at the blood?
No. It turns out that's not what we can find is memory. We can see if
Yeah.
there's a cellular response and that T cell memory has been developed, whether it's from wild type or from vaccination. And in fact, it's easier to find out dogs that have been infected naturally. They get a much bigger response, probably because they had a lot more disease that they got over. And we're doing we're taking 10,000 spores, but then they don't keep propagating afterwards. And so it's probably a much smaller fungal burden and much less stimulus. And yet it still produces protection. So,
Protection for life, it sounds like.
right. But you'd know if you hadn't been vaccinated, um, um, you'd know that.
Mhm.
You wouldn't get it. You wouldn't be silently vaccinated. So if the test was positive and the dog you knew hadn't been vaccinated yet, that would be from past infection. Or it probably wouldn't make sense to do the test first. It's probably easier and probably cheaper to just go ahead and get the dog vaccinated, and then if you want to know, do the test afterward. And if it shows protection or shows the memories there, you won't know whether it was because of a previous infection or the vaccine. You'd just be feeling comfortable that it's got it.
So that would be a titer that they're running to find
No, the
that out.
titers, well, we actually have a test that shows antibodies too, but the commercial tests don't show a vaccination. They're
Oh,
insensitive to the vaccine. Again, I think because it's such a small amount of infection compared to what you get with a naturally acquired infection.
okay. So it's a different test, a different blood test to determine protection from with the vaccine.
That's right.
Oh, interesting stuff. So, do you have any other studies going on besides the
Well,
vaccine studies?
we do.
Probably a lot.
We have interest in many different directions with valley fever. It turns out that veterinarians are really, at least in the endemic regions, are really good about looking for valley fever. If something doesn't look right, you can tell me, Pamela, if this is correct. If something doesn't look right, valley fever is on the differential.
Exactly. It's on the differential in an endemic region, period.
Right. And that's not for human doctors. It's been amazingly mislooked for.
Mhm.
In urgent care, and I've been working with Banner Health Urgent Care because the University of Arizona practice is now affiliated with Banner Health, which has got a very large presence in Arizona. And urgent care clinics, of which there are 55 or so mostly in Maricopa or around Phoenix area, they in 2020 were testing for valley fever if a patient had pneumonia 2% of the time. And I've working with them and with
Wow.
various things. We've now gotten that up close to 50% of the time. And I'm very proud of
Should
that.
be
And I think,
100.
well, it turns out that there's reasons to think it shouldn't be. I mean, some patients refuse to be tested.
Oh, wow.
Um, and then there may be other reasons. And then there's
Yeah.
there's the problem with coding and whether or not the patient really had pneumonia and the code says they had pneumonia. So I would be content with more like 70% or 80. But that would be huge. And
Yeah.
we're working on that very hard.
Are
That's
they
one
using
area.
that rapid test that they can check within like 30 minutes in the urgent
That
care?
would be a game changer. Yeah, that would be wonderful. There's interest in that. There is a commercial test that's made by a company in Oklahoma, which it's not something for the clinic. It's a CLIA or it's a reference lab does the test because it's too complicated. It's not just a dipstick. It is a dipstick, but you have to prepare the specimen before you do the dip. And that's a 1 hour test. And if something like that could be done in the clinic, that would be huge. And there's interest in that. There's some friends of ours at Arizona State University that has interest in doing that, and hopefully that'll emerge. Because if we could get a diagnosis before the patient leaves the office or the veterinary clinic, you'd be
Yep.
much better along.
Yep. The
Um,
faster you can diagnose it, the faster they can get treated and the better prognosis they have,
right.
for
And
sure.
then And there's, as you know, the treatments for valley fever are not one day treatments. They're many months, and some people, as I said, get treated for
Years.
life. Dogs too, and so
Yeah.
that's because none of the treatments we have really cure this disease. It suppresses the fungus, and so there's a need for a curative treatment. And actually, there's some candidates coming along, and the Valley Fever Center is involved with some of those clinical trials to see if one of them might be a cure.
Is
Um,
nicomycin Z still on the board for studying, or has that been scrapped?
No, it has not gone further because of money.
Yeah.
It's licensed to Valley Fever Solutions. I was a part of creating Valley Fever Solutions. Full disclosure, I'm no longer on the board or get any benefit from that. But Valley Fever Solutions still exists, and they would love to develop the drug, but it needs the funding to do so. And again, anyone who has an opportunity wants to get their name on a drug, here's one that would be a wonderful choice, which is potentially a cure for this disease.
Yeah. Isn't that one fungicidal? It
Could
kills the
be.
fungus. Yeah.
It's potentially fungicidal. That's what the preclinical data looks like.
Yeah. We need more money to go into research on the drugs that can cure valley fever for sure. Wow.
And then there's this whole area of the fungus in the environment. I mentioned the um, the models for where the endemic regions would be based on temperature and rainfall predictions over the century. But it's as the wind blowing back and forth between Arizona and California suggests, it's probably more complicated than that. And it may involve intermediate hosts like rodents in the environment, and other. It could have to do with the micro environment in the soil. Many things we don't really understand about that. And the Arizona Board of Regents funded some work over the last 3 years, which myself, and collaborators at Northern Arizona University and also ASU, we've been working together to try to understand that ecology much better. And it's been very productive. One of the things that came out of that was we collected over 800 specimens of coccidioides from humans, and then did the genomic sequence of all those strains. And of those, we found 16 clusters where the genome was identical in
Wow.
each of those clusters, suggesting that probably those individuals in those clusters got the infection from the same source. And working with the Arizona Department of Health Services, they're going to do some surveillance and see if they can find out possibly where that source might be. So there's lots of interesting things to do with it before the infection starts. Also, that might lead to some interesting ideas for control.
Oh my goodness. Yeah, I think some of those outbreaks were in California after a music festival or something, and then the archaeological site in Utah. I think that was another one.
Right. And in the foothills of California, I think it was one of the state colleges from California had some wonderful outbreaks in archaeology sites. Um, and the there've been some Tennessee Marines, I think, reservists that went out on a reserve thing in California and all came back to Tennessee, and half of them got sick. And nice outbreaks like that.
Wow. Yeah, they probably got a big blast of fungus from whatever they were doing.
There was a famous outbreak after an earthquake in Northridge in California,
Oh, wow.
downwind from that earthquake.
Oh, so they're trying to figure out all the environmental risks and then how do we prevent that? How do you prevent, going out in the wind and the dust or, going places? That's a tough
Yeah.
one.
The actual risk, at least for humans, the actual annual risk is about 4% per year. And we know that if you try to find the valley fever fungus in the soil, or even where we think of it as very endemic, it's most of the place it's not. It's in very
Pretty
small
spotty.
parts of the acreage.
Yeah.
So, so in addition to the idea of the map getting larger all the way to Canada, for example, it's also possible that with climate change, we might find that the intensity of the infection will even get larger here in Arizona or in California, and where we already know it has a foothold. And that could make for profound differences. I mean, now it's, I said one chance in 25 a year. The median time of residence is 12 years before people get diagnosed with valley fever. So it's not every day that you get infected. But what if climate change to us made that be half that, or you know,
Yeah.
or twice as much, or three times as much? It'd be a totally different ballgame.
So it's going to increase the rate of infection in the endemic areas and then increase the scope of infection across the United States with the change in climate. Are they working on anything with people's immune systems? Like it seems like you would have your if you're immunocompetent, you'd have a better chance of not getting it versus immunocompromised. Are they doing any research on that?
We are. Um, so, so yes, if you're immunocompromised, like if you have AIDS or an organ transplant or some very profound immunodeficiency, you're much more susceptible to complications of valley fever. But it turns out if you look at of the people who get infected, the vast majority are not immunocompromised.
Hmm.
It's mostly normal people, immunologically normal people. And we don't see families. So it's not like sickle cell anemia, where that's due to a single gene, which is called Mendelian inheritance after Mendel. It's what's called complex genetics. Now, for me, all genetics is complicated, but
Yeah.
technically, complex genetics means the participation of two or more genes cooperating to develop the risk. And some work we've done with collaborators at the National Institutes for Health, particularly Amy Shu is a very, very astute scientist. We've identified that much of the risk of getting the complicated valley fever problem is actually not so much the T cell itself, but the pathways of the innate immune response leading to programming the T cell, or the immunity of the T cell. It seems like the early events before it gets to the memory cells that accounts for most of the problems, or where the success has to be, and where two or more genes in combination seem to disrupt that success. And we're very interested in that. And that, if in fact, some work we've done with Yves Lucier, who's now in Utah, very, very interesting statistician, used to be a pulmonary physician by training. We've actually identified a small number of roughly 55 genes which are associated with their expression is associated with disseminated disease. And that
Wow.
finding really is intriguing, suggesting we might actually have a test, make a test, or could make a test that would tell people if you moved to Arizona and you got infected, what your risk of getting a complication would be. And that has a prediction, ideas for, well, A, should I move to Arizona, or is there some way I should be treated differently if I were to get infected?
Wow. So they're looking at the genetics involved in your predisposition to getting sick and
That's
then
right.
how to um, change that. Are they looking into, how to change the genes so that we're
Well,
not
that might be. There are a couple of genes that are of interest, but we're getting down the road a bit. I think we're not gene editing yet for this disease. It's theoretically a possibility. Things would have to become much simpler before that would become a reality, I think.
how about epigenetics? Have are there studies on, just lifestyle factors that are different between the people that are getting really bad disease and not with
Yeah,
our healthy people that are getting infected or healthy people?
that's a great question too. And we've talked about that. I don't know that there's hard data for that, but it's certainly a possibility.
Yeah.
And there are populations who seem to be at increased risk, and we don't quite understand why they are. And it's possible that epigenetics could play a role in that. Um, and it's also possible, I've understated the possibility that there are differences between strains of the fungus.
Mm.
They're really quite similar. If you look genomically at the different strains that have been sequenced, they're very similar, but there are differences. And it's also possible that some strains are more virulent than others.
Oh,
Um,
it
and
depends
that's
on the
that,
strain.
the data for that is not yet solid, but that's a possibility. I'd say that it's probably not been looked at very carefully. It's really the focus. And part of it is that if it looks, some of the isolates that we have used in the lab for a long period of time that are very virulent mice came from people who had very avirulent or uncomplicated infections.
Hmm.
So it doesn't seem like virulence in one species necessarily means virulence in another. So it's complicated.
Yep, it sure is. So you did another study on managing cavitary uh, coccidiomycosis. Um, and
Right.
and was that to help clinicians know how to treat people with advanced or disseminated valley fever?
So I haven't talked about that much. In addition to the complications where the fungus goes through the bloodstream to other parts of the body, some people develop a pneumonia that doesn't go away, that it forms it causes destruction of the lung, and that leads to areas where we call them cavities, where there's just no lung there anymore. And that's really a different, it turns out that's a different group of people. The people who get that complication, almost none of them have disseminated disease, and vice
Oh,
versa. People with disseminated disease typically have pulmonary infections that kind of get better. And and there's really at this point yet to be seen any genetic association with the chronic pulmonary problems. Diabetics are more likely to have problems with chronic cavitary cocci, but other than that, not much in the way of human comorbidities or genetics has been really identified with it. So they're sort of a different problem, and it becomes a local problem that's managed very differently than the destructive lesions outside of the lungs, and sometimes is helped by surgeons taking the cavity out. Sometimes it's too extensive for that. It can be a difficult problem too, but just very different than the disseminated disease.
wow. So you had done that study to help um, figure out for clinicians how to manage those.
Right. I was part of a consensus group that discussed the sort of the spectrum of approaches to that, and it's very individualized. It
Yeah.
turns out, and there's regional differences. And then when you talk about doing surgery, that depends on the experience and the specific um, practice patterns of the surgeon, he himself or herself, and um, it's it's very individualized, and so having a comprehensive attempt to try to instruct people how to take care of it, would you end up with some suggested um, approaches, but then ultimately individual patients get managed in what seems best for them.
Yep. It's all everyone's different when it comes to valley fever. So that type of valley fever is not disseminated then. That's still in the lung.
That's right.
Okay. Okay.
And
It's
then
just
there's a lot of symptoms. There are a
there.
lot of symptoms in humans, at least, like rashes and aches and pains. Fatigue is a very common symptom in humans. None of that, even though it's your whole body gets involved with those things, that's really not disseminated disease. And people sometimes are confused by that. Even though you have a rash, that's more the immune response that's triggered by the infection rather than there being fungus in the skin that's causing that rash. Now
Mhm.
you can have the fungus in skin lesions, but that's where it's disseminated disease. Most people who get that stuff gets better on their own, and it heals without really any damage at all.
Nice. Yep. Yeah, it's confusing between dissemination and then just your your regular effects of having valley fever with the aches and pains and rashes.
Right.
Yeah,
Which can go on for such a long time. People get very annoyed by it
I bet.
and
I,
frustrated.
yep. Yep. I've had valley fever a couple times. Have you, all right, and I probably still do have it. Yeah. Have you had it?
Yes. I didn't know I had it until I did a skin test back in 1983 and
Yeah.
discovered I must have been infected.
Wow. Is that what got you started with valley fever?
No, I at Stanford when I was doing my fellowship work, I worked in a laboratory that worked on several different fungi, and so I had an interest in valley fever, but I had an interest in other things. But when I took my first and only job at the University of Arizona back in 1978, once I took that decision, made that decision, it became obvious that the thing I should be starting to pay more attention to was coccidioides.
Absolutely.
And I've no regrets. It's been, as I say, quite addictive.
Yes, it's a very unique fungus. Well, I really appreciate you sitting down and talking with us today
Sure.
and giving us a wonderful presentation on valley fever and really some important information and things that are new to me and new to I'm sure a lot of people. And the vaccine is really exciting. I'm so glad that that may be coming out this year for dogs.
I'm very excited about it.
Yeah.
It would be the first vaccine ever for any fungus if we can pull this off.
Wow, that's interesting. It's it needs to be done. Well, thank you, Dr. Galgiani.
Well, it's a
And
pleasure, and thank you very much for being interested in this problem.
yes, yes, it's we need to spread awareness about valley fever and the more we know, the more we can do and we can prevent it.
And as you said, there's lots of information on our website, which I think you're going to post, right?
Yes, I will post the link to the Valley Fever Center for Excellence and I'll post a link to the vaccine information also.
Great.
All right.
Thank
Thanks.
you very much, Pamela.
Thank you, Dr. Galgiani.